By utilising AI-powered tools first deployed in drug discovery, Quercus Biosolutions reckons we can finally deliver the kind of efficacy we expect from chemical crop inputs to the biological sector.
What we're really doing is using the toolkit that's been developed for protein structure and function prediction that was really the object of the 2024 Chemical Nobel Prize to build novel proteins from scratch to target specific sites of action that would have the effect in crop protection that we want. We think that's equally valuable for really developing a protein biological that's useful as panning nature. And we've chosen to focus in an area, weed control, where nature doesn't actually have a lot of starting points for a protein herbicide. We really started with two key problems in weed control. The first problem is all the sites of action that we're going to be interested in are inside of plants. They're usually in plant cells. They're often in organelles within plants, and sometimes they're only active in certain particular special tissues in the plants. So we had to actually build proteins that can enter and move within a plant, and then we had to target things that we thought would create a herbicidal activity. To really minimize the variable space, we chose to first look at existing sites of action so that we could get a proof of concept result and show this approach can work. Importantly, because protein interactions can be quite different than a protein small molecule interaction, we can develop proteins that will have a different mechanism of inhibition at an existing site of action. So we might actually be able to control, for instance, resistant weeds straight away with a protein inhibitor where there's already a chemical site of action and a chemical inhibitor that has resistant weeds. So my co-founder, Matt Crisp, and I really looked across the sector, but we felt that the most richly funded area, human medicine, would have the most advanced application of these technologies. And we looked at really more than 10 potential partners in that space and did deep dives with 3. We identified our partner Ordeos really through a deep investigation and as well as an ability to align incentives. They've been building their platform for pharmaceutical design proteins for 5 years. They've brought in over $20 million of investment, so they're really quite far along. And the fact that we've made so much progress since our founding less than 2 years ago is in part because of the capabilities that their platform has brought for us.
So as I understand it, you're not just looking at, when you're trying to design these proteins, it's not just about mode of action and obviously efficacy, but factoring in practical constraints from the outset, including things like manufacturability.
Yeah, that's exactly right. We see the AI both as really highly impactful in the discovery phase, where of course the first question is, can we target a site of action? Can we inhibit its activity? But equally in the things we would usually address in early and even late development or even regulatory, the platform at Ordeos can handle multi-factor constraint-based optimization against many design objectives. And so we can ask for strong micromolar inhibitors of a given target, but we can also ask that it be less than 5 kD, as in small, and therefore somewhat easier to manufacture. We can ask for stability. We can screen against non-target organism impacts. In some CP categories, that's especially important. So we actually see the process we're building for these new designed protein crop protectants as a process that's going to learn and get faster and faster with sort of every new challenge we tackle. I mean, I think that, the linear process is what's given us 95% of what we buy in crop protection today, right? The chemical small molecules that are so used, they're, solid performers, they are cost effective, and they have a dominant share in the market. They've all come from that linear process. Now, there are some great innovations, both at big companies and some of the folks here at trying to enhance that process. But when you defer those, what I sometimes call them second or third order properties, when you don't address those until 3 or 4 or 5 years into your process, of course you're sometimes going to fail. And the reality is, in that chemical process, something like 99% of what gets discovered doesn't make it through the process. We want to pull as much of that forward as possible, as well as continue to learn and teach our AI from those downstream sort of development challenges, from those downstream potentially regulatory challenges. We think we can pull all that forward. And I think that opportunity, creates a dynamic where honestly, we could look ahead 10 to 15 years and half the crop protection segment could be these types of molecules. chemicals. Our first really sort of minimum viable product idea is simply a broad-acting herbicide, so broad spectrum, controls lots and lots of weeds. Importantly, would control weeds that are resistant to some of our major chemicals today, like glyphosate and some of the other chemical solutions, and could just be used pre-plant and would work the way Roundup did 30 years ago, before Roundup started to really have sort of economic level resistance occurring in the field. We think that's a great product. We're actually working on targets that can deliver that today, and we've achieved initial proof of concept results. We've built first in the world designed protein herbicides that show activity on weeds that is very similar in terms of a molecule per molecule activity basis with chemicals that target that same site of action. One of the things that's exciting about the biological category that lots of the businesses that are here in biologicals are talking about is in many jurisdictions, the US is one, Latin America has some very good jurisdictions, we can come to the market in the US, well-established precedent at EPA that the EPA biopesticide registration framework is really what we can use to bring these products to market. That's less than 2 years, less than $2 million of investment. So in addition to partnering with the big businesses where I've spent half my career, like Corteva and Bayer, we can also partner elsewhere in the chain to bring these solutions forward. And we think that's really important for us as a business because it expands the opportunity set beyond just the perhaps 5 companies that might be discovering new chemistry solutions today. We think mini proteins are really excited, and I see proteins in general and small design proteins specifically at really sitting in a great space that's between, of course, chemical crop protection, 95% of the market, strong COGS, it works, it's consistent. Now they have their challenges, but they really mostly are on timeline. And biologicals that can be more complex, where we go out and we find an exciting organism in nature, maybe a mycorrhizal fungus, maybe some interesting bacteria. When you go all the way to an organismal solution, I think you find amazing things because nature does amazing things, but turning that into a product can be really challenging. You've got a domestication problem. Nature often doesn't think a lot about cost of goods as it builds solutions. It really doesn't. Those mycorrhizal fungi are getting a free ride from their plant partners. Plants don't worry much about energy. They have the opposite problems. So proteins are a defined molecular entity, just like a chemical, but they have many of the benefits, both actual environmental benefits and regulatory advantages of a biological. And we think it's a great segment and a great opportunity.